on. I'm not sure. I don't know if you need to be able to see me, but anyway, before we begin, I would just like to acknowledge the Chumash people, who are the traditional custodians of the land on which NCEAS is located. I'd like to pay our respects to the Chumash elders, past, present, and future, who call this place their home. As we come together today to discuss the future of our work, we recognize that the Chumash people and so many other indigenous people around the world are still fighting to dismantle the legacies of settler colonialism in their tribal territories. So over the last few weeks, it's been really fun getting to know folks through some of the pre-workshop sessions that we did and the brainstorming activities. And for those of you who did the Compass training, I got to do all three of them, and it was really fun, really exciting. And we look forward to continuing this engagement over the next couple days. We really welcome, encourage open conversation and respect for fellow participants. So we really want to create an inclusive, welcoming, supportive environment where all ideas and all people are welcome. So we really strongly encourage you to adhere to that. We have two overarching goals for our time together, which I hope is clear through some of the materials we've been sending around. The first is to create an informed and inclusive research agenda for the next decade of synthesis science and ecology. So how can we help shape the direction that our disciplines are going with the work that we do? And second, really foster a growing network and opportunities for collaboration with familiar faces or new folks that you meet through this venue. So really encourage both of those. So for that first goal, we'll be spending the next 2 days together hearing some really inspiring research and directions people are currently taking, and then working through some small group exercises and to collaboratively design a research agenda for the future. So we hope that the plenary sessions help inspire and create expansive thinking. And then in the breakout sessions, you really dive deep into helping flesh out some of those ideas. And we plan to ultimately produce a manuscript that is dedicated to the process and the outcomes of this workshop, and all who are interested will be included as co-authors on that paper, so more to come on that. And for the second goal, obviously it would have been lovely to host everyone in Santa Barbara. That was the original plan, but we are trying to make this virtual experience as exciting and rewarding as possible. We encourage you to use the network timings that we've set aside and the lounge spaces to connect with people, discuss new ideas, pose ideas for potential spin-off collaborations, however you want to use it. If we were in person, you'd be meeting in the hallways or at the coffee stand or over a meal. And I know we don't get quite that in this experience, but hopefully you can take advantage of those spaces and those opportunities to really try to make the most of this virtual experience we're going through. There's lots of modes for engaging in the platform. We also encourage you to use the hashtag #FutureOfSynthesis to tweet about what's going on and to share a dialogue there, to encourage others who are not part of this direct workshop to participate through that mechanism as well. And I'm going to turn it over now to Heather, our program manager, who's going to walk you through some of the logistics around the event and how we can effectively share our ideas moving forward. So Heather.
Great, thanks, Ben. And a big congratulations to everyone that successfully navigated to this room. Pat on your back. And I know that, there were some different difficulties with links, but why are we using Keynote Chat in the first place? And the big reason is that we're hoping that it does mimic some of that live interaction that you have. And once you get used to the platform, we really think that it does feel like you can move from one room to another, which, will be a little bit different in our virtual world. So just, a pro tip, during these plenary sessions, we will be in a Zoom webinar, which means that we can't see your videos and you will all be muted, but we do encourage you to use the Q&A function and the chat function after when the speakers are speaking. But after we're done with the plenary sessions, you'll notice in Keynote Chat that there's a list of rooms listed down the side. And that is when you can freely move into these different breakout rooms. And so I highly encourage you to have both the Keynote chat window open and a Zoom window open. And I know that can get difficult if you're working only from a laptop. But that way you can see the collaborative notes, which everyone should have access to type into these notes while you're in a breakout. And you'll also be joining the Zoom call so you can see each other. We do ask that, try and share your video if you are comfortable to make it feel a little bit more like we are together. And maybe mute yourself while you're not speaking, just because there might be a lot of background noise. I know I have a dog that likes to randomly bark. We have built breaks into this agenda. We understand that this is a long day in front of a screen, but please feel free to, do what you need to do, get up and stretch and take the time that you need. And as Ben mentioned, one of our goals is to connect and collaborate. So some of you were able to make it into the lounge this morning, which is great. That lounge space will be open. It's open now. You can always go back to that lounge space, but it will be there during lunch, during the breaks. And to get to the lounge space, you go to the top of your Keynote chat. If you're in this main room now, you go to the little blue arrow that's next to day one, which will take you back to the home page. And from the home page, you'll see lounge. But if you, when in doubt, you can come back to this main page and you will see in the notes in bright yellow kind of where we are in this world of Keynote chat. And we also, I wanted to point out on the home page, if you click on the menu, which is the universal 3 bars across menu, you'll see something called resources. And some of you have already started to populate this, but if there's articles you would like to share, or if there's a conversation you want to get started, you can post a question or an article as a resource and encourage people to keep the conversation going there. So I would recommend to check out the resources, during one of your breaks or during another session. And so if you have any technical difficulties, you can reach out to Courtney. Her email is listed on the notes of this main page. There's also a chat in the lower right, which we will try and pay attention to. And feel free to reach out to us and we will address the questions as they come up. And so with that, I would love to turn it back over to Ben to get us started on what synthesis has looked like and where we're hoping it can go.
Great. I'm just going to share my screen now. Heather, can you see my screen?
I can. Looks great.
Great. Welcome, everyone. So I want to give an extremely brief history of ecological synthesis to just set the stage for where we've come in the last 25 years or so to this point in time, and how that might anchor or position the way we think about moving forward, or how it might encourage us to pivot in new directions. So I want to start with a little bit of reminder of where things were 25 years ago in terms of the digital society that we were experiencing. For those of you who were born or were still a child in 1995, you may not know this, or at least not remember it. But this is when AOL, America Online, just started in 1995. Only 10% of Americans were on the internet, so a really small percentage. Dial-up modem access was still the norm. Email was really just starting to take off. Not that many people were using email. And this strange thing called a zip drive had just been invented, and it was a really big deal in terms of the amount of data you could store on it instead of the old floppy disks. This is what homepages and websites looked like. This is not something you would hopefully see anymore, but people were excited to be able to create this digital vision of themselves, and were playing around with all sorts of interesting ways of representing that. Here's another example. Jesse was really excited to share his love of skiing And it was very common to like put up a website that was still under construction. And there was an unbelievable number of GIFs and icons you'd use to say that your website was still under construction, which is hard to imagine these days. Ecology in this time was also digitally challenged as everything else was back then. Presentations were given on these slide carousels. I gave many myself using these. You would take these to conferences. This is a figure from the journal Ecology in 1995, which is like hard to even read, but this is the state of visual display back then. And importantly, the culture and practice of ecology really was not that supportive of the idea of data reuse. It was really the idea that you needed to go collect new data in order to test ideas rather than reuse existing data. And also similarly, the idea of sharing data or ideas was really rare because these were seen as the currency of success in academia, and you didn't want to share that. It's how you helped your own career move forward, which by keeping hold of your own ideas and your own data. But with really great foresight and a lot of community input, NSF funded the first synthesis center, NCEAS, in 1995 to really try to break through this cultural setting and shift the approach and narrative around how ecology could advance by embracing synthesis as one of the tools that we use for research. Since 1995, there's been about 1 million papers in ecology and environmental science across all the journals. And of this, about 1 to 2% of them have been synthesis papers. So not a lot, but certainly an important contribution to this 1 million papers in ecology. Just to make sure we're all on the same page, this is how I think about what synthesis means. And of course, there's many ways of thinking about it, but we really think about how we analyze and integrate disparate data, methods, tools, theories, and knowledge to provide new insight. So there's many ways to do synthesis, many approaches, many pieces of information, many ideas that can be brought to bear in order to gain this new insight. Just an example of the kinds of things that can come from synthesis, I went and found the top cited paper from each of the 4 synthesis centers that NSF has funded. So there's the value of the world's ecosystem services and natural capital that came out of NCEAS, the Bayesian phylogenetics with beauty and the beast that came out of NESCENT, the Evolution Center, why interspecific trait variation matters in community ecology that came out of NIMBIOS, and then the capacity shortfalls hinder the performance of marine protected areas globally that came out of SUCCINCT. So you can see really interesting scope of the kinds of questions that these synthesis centers and the researchers in them are tackling. And of course, this is just one of the thousands of papers that have, or 4 of the thousands of papers that have come out of the synthesis centers. Now, work we did last year, we looked at the influence of synthesis on the field, found out that on average, synthesis papers are cited 5 times more often than other papers in ecology, which is just only one, but it is one metric of the influence that synthesis can have on our discipline. But more importantly, we found that synthesis can be a really powerful tool for discovery in helping bring resolution to debate when you've got multiple ideas across tens or hundreds of studies. You can bring them together to find what the kind of the overarching narrative or story is across that work. It can help redirect the focus to new topics by showing where we know things, where we don't know things, and of course in doing so can launch new areas of inquiry, both by finding those gaps, but also by bringing different disciplines or data sets together that haven't been done before to kind of open up new areas of research. We looked at the collection of papers that have done synthesis, this case out of NCEAS as a case study, but it was really trying to get a sense of what synthesis writ large is doing. And you can see these interesting clustering patterns that are done by topics that keywords of the papers highlight. And so the topics themselves kind of show the focal areas of synthesis research in the last 25 years, but also interesting all of the connections between them where papers are linked through keywords to each other to show kind of the shared but also interdisciplinary nature of many of the papers that are being studied, or the research that's being studied in these papers through synthesis. And in fact, we looked at some network metrics of these collection of papers and found that on average, the kind of the diversity of topics or themes that are being used or studied in these synthesis papers and the connections among them, these diversity and bridging metrics, are much, much higher than the control non-synthesis papers, really speaking to the interdisciplinary and expansive topics that synthesis tends to do compared to other approaches to doing ecology. And as I said, it also really is an important tool for identifying gaps in what we've done. I think this is something that often gets overlooked with synthesis. It is a very powerful tool for getting that bigger picture of what's going on by pulling information and data and ideas across different studies and different disciplines together. But it also really is powerful in helping identify the gaps. So you can cluster and find the similarity in these groups of marbles or research that's been done. You can classify a bunch of other marbles and other ways by doing synthesis, but maybe you haven't looked at this other set of marbles. And by doing synthesis, it really highlights which marbles you have studied and which ones you've left behind. And that can help redirect and focus energy on areas that are understudied and need more attention. I'm really excited about what the next 25 years will bring of synthesis and ecology. We really have the potential to leverage really exciting new tools and resources in what's I really think of as sort of a data revolution. We have huge amounts of new data, very large data coming online. We have these standardized replicated data that are being produced through things like NEON or other coordinated monitoring efforts that provide a whole new set of kinds of data that can test new ideas. And we're getting access to traditional archives like in museums that are being digitized and allowing much, much greater access to these resources and not requiring you to travel to the museum to find them, for example. So I think we're really in kind of a new era, or in the middle of the emerging new era of data availability that is really going to transform the opportunities we have in synthesis in a way that shifts from that AOL dial-up modem era 25 years ago to today, saw huge shifts in the last 25 years. So I'm really excited about what the future holds for us. Of course, it's not just the data assets, but also the computational tools and cyber infrastructure that's being brought to bear on helping us access, process, analyze, share all of these data. And this infrastructure is really accelerating the pace at which we can do synthesis. So, work that in the past maybe took hundreds, if not thousands of hours to discover, clean, compile, standardize, and analyze data, scattered across really diverse and globally distributed sites. We can now do that potentially in just a few days by a single well-trained researcher because of this cyber infrastructure that's in place to allow us to access and analyze the data. I also think we are recognizing the power and the need for greater diversity in ecology and synthesis in ecology, both in the people and expanding the people that are part of the ecological endeavor and synthesis within that, so more diverse ecologists, and also the disciplines that are brought to bear. The modern environmental questions really are demanding expertise of many disciplines that we bring together with ecology to help us better understand the planet that we're living in. So for the next 2 days, I hope that you feel inspired, that we find questions and topics that are ripe for synthesis, where there are gaps in the areas of research that we can help fill, where are blind spots that we need to be paying more attention to, and really, what does the future of synthesis look like? And to connect, make new colleagues and friends, find people with similar or even more important, different interests that you can stay connected to and potentially spawn new collaborations with, or if nothing else, just make a new friend. And of course, build our community, our discipline, and run with that. If there are ideas that emerge from this workshop that you want to pursue, go for it. Please, let's use this opportunity the next 2 days as a launch pad, not as an endpoint, and see where we get to. And so, ending where we begin, I'm really excited about the future of synthesis and ecology. It is definitely an endeavor that's under construction, and excited for the next 2 days. So thank you.
Thanks, Ben. And with that, I think we would love to move on to our first speaker, who we have Dr. Kristy Kroeker from the University of California, Santa Cruz. And she will be talking to us about how we can advance our understanding of the cumulative impacts of multiple stressors and the emergent effects of global change. So with that, I'll invite Kristy to go ahead and share her slides.
Okay, are you guys seeing that?
Yes, looks great.
Okay, great. Sorry for the little delay there. So first I wanted to say thank you to the organizing committee for having me here. It's a pleasure. I do very much wish I was in Santa Barbara right now with all of you, but I will stay here in my little office and do my best. And I'm looking forward to interacting with all of you over the next couple of days. So to give you a little bit of background on who I am an associate professor at the University of California, Santa Cruz, and I'm a marine community ecologist. My work is really, I use a number of different approaches to try to address the community and ecosystem responses to global change in the ocean. So my title is a little bit different than what I sent in, but this idea of scaling up in global change biology is really close to my heart and always in my mind. So let's see. Okay, so as a community ecologist, I have always been interested in how we go from this understanding of organismal responses to global change, which is where a vast majority of our understanding lies, to putting this organism in the context of its community or ecosystem. So I really want to know how the interactions in this really dynamic functioning ecosystem affect not only that fish, but also how the interactions between the fish and the sea star and the macroalgae scale up to affect the properties and the services from our ecosystems. Now, this is challenging. The idea of scaling up might be impossible in and of itself, but I'm going to talk about some of the challenges and where I think synthesis might play a role. Before I get into those, I just want to talk briefly about global change in the ocean for those of you who don't spend your time underwater. Global change in the ocean is really threefold, at least for this talk. I'll be talking about 3 issues. So the oceans have absorbed over 90% of the heat caused by global warming. Due to decreases in oxygen solubility, that warming is leading to deoxygenation across the world's ocean. At the same time, the oceans have absorbed over 25% of the carbon dioxide that we've emitted since the Industrial Revolution, leading to this process of ocean acidification. Now, one of the big challenges, I think, for scaling up is the difference between how we actually collect our data, or the empirical data we have on organismal responses to global change, and how global change is actually going to play out in nature. So this slide is showing the results from a meta-analysis that I did with some colleagues a number of years ago now, looking at the effect of near future acidification on the growth of a range of taxa, which I have organized down on the bottom. So I'm using the natural log response ratio here, so things below 0 mean a reduction in growth in response to near future acidification, and vice versa above 0. Now, I show this because the vast majority of these studies, 96% are actually using a single species in a tank and bubbling that tank with carbon dioxide to create ocean acidification. Now, the first challenge is that we know that species interact. So as a community ecologist, we all know this, that species interact. And what research has been done on community and ecosystem responses to global change suggests that these species interactions strongly mediate the effects. So it brings into question, well, how useful is that organismal information when we try to scale up? And then the second major challenge I'm going to talk about is that stressors also interact. So I introduced that global change is, this multivariate problem. Here I'm showing you the results of another meta-analysis. This is about 125 studies where they factorially manipulated warming and acidification, and then looked at primarily organismal responses. We calculated interactive effect using the natural log response ratio, and then categorized them into these three categories of either additive, synergistic, or antagonistic. And I'll just say that broadly, I'm defining synergistic right now as warming exacerbating the effects of acidification or vice versa, and antagonistic being opposite, so they're actually offsetting the effects. But the big picture that I want you to see here is that we generally are seeing additive, synergistic, and antagonistic responses across the really wide variety of studies that are looking at the combined effects of these two drivers or stressors. And this is not particularly useful for making predictions. So I'm not going to get too down in the weeds today. I'm going to give you a couple examples of work that my lab is doing on these challenges. But what I hope you take home is really more about this idea of where does synthesis fit in, and what is the future of synthesis in particular in this idea of scaling up global change biology. So I'll start here with this idea that probably everyone is very familiar with in their intro ecology course, where observations and experiments feed back on one another. And synthesis has a really important role in this ecosystem. It's very obvious that our observations and our experiments feed into synthesis, but I think it's less well appreciated how synthesis needs to feed back into our observations and experiments. And I would say or argue that really to move forward in this field, we need all three, that we can't generalize just through the observations and experiments that have already been done, but that the feedback between these three fields is really going to lead to the most insight. Okay, so I'm going to focus on just two examples, one on the species interactions and one on the stressors interactions. Again, you don't need to follow too much into the details of what I'm talking about if this isn't your field, although I'm happy to talk about it. But again, I hope that they provide some insight into this idea of feedbacks between the different disciplines. So starting with species interactions. So trophic interactions have been highlighted as a really important way of thinking about scaling up, in part because we know trophic interactions can play really key roles in structuring communities and ecosystems. And I have been interested in a number of years of, can we switch from this really organismal focused view of trophic interactions to one that thinks more about the rates and processes, or how global change is going to affect rates and processes. Now, this is also the focus of some work. This is a meta-analysis done by Ivan Nagelkerken and Sean Connell a number of years ago now, published in PNAS, where they synthesized all of these studies that looked at the combined effects of ocean acidification and warming on key rates and processes, and then applied them into this broader model that I'm showing on the right. This is, a really beautiful infographic about how primary production, consumption, secondary consumption are going to change, and how that scales up to influence species abundances, or things like fisheries production, resilience, or ecosystem services. Now on the left, I'm just showing one little piece of the meta-analysis, and this is their synthesis of the effects of ocean acidification. T stands for warming or temperature, and then ocean acidification and warming combined on predation rates. And you'll see that across these three different treatments, they generally found that global change was likely to increase predation rates. Now, I think that this is super important work, but that there's complexity in here that we really need to embrace when we start thinking about scaling up, and that we need to be somewhat careful about our generalizations. Instead, think about synthesis in this broader ecosystem. So I'm going to talk about really briefly some results from a meta-analysis that I did in collaboration with Ty Kingdinger and Jason Toy on this same idea of the effects of warming and acidification on trophic interactions. We used a slightly different effect size, and we had a higher number of studies because this was a few years later. But importantly, we looked at the variation in those effects among taxa, life stages, and which trophic level was treated. So I'm just going to jump quickly to our results. So here I'm showing you that same figure from Nagel-Perkin and Connell that was on 2 slides ago, looking at the effects on predation rate, and then our comparable data. So again, we're using a slightly different effect size, and we have a lot more studies at this point. Unfortunately, our colors are slightly different, but I have them lined up in the same way. So ocean acidification, warming, and then ocean acidification and warming combined. And you'll see that qualitatively, our results are quite similar. So we don't detect an effect of ocean acidification on predation rates, but we see increases in predation rates in those warming and acidification combined treatments. Now, I think what I'm going to talk about next is what I think is some of the really important nuance in this data, though. So here I'm going to take that same figure that was just on the previous slide, and then I'm going to break it down into studies that treated only the predator, only the prey, or both the predator and prey to those environmental conditions. And what you'll see, starting with acidification, is that we actually do see a decrease in predation rate in the acidified treatments. But when both the predator and prey are treated, we don't detect any change in predation rates. Now, importantly, this is actually repeated in the warming-only treatments. So the significant increases in predation rates were in studies where only the predator was exposed to warming. And when both the predator and the prey are exposed, we are not able to detect an effect. And again, the same thing is true in the acidification and warming combined treatment. So what does this mean? None of the studies in this meta-analysis went out to test whether the individual, if you only treat a predator or only treat a prey, or you treat both of them, how the results differ. But by looking at the complexity in this data set, our results are suggesting that the most ecologically realistic conditions, when both predator and prey will be in future change or global change conditions, we don't see as strong of effects. So coming back to this kind of observation, synthesis, experimental conceptual framework that I brought together, what I hope I just showed you is that by actually embracing the variability, rather than always shooting for generality, we can come up with new questions. And that those questions may not be answered fully by synthesis, but they bring us back to our observational and our experimental work, which can refine our understanding and then again push the synthesis forward. Okay, so now I'm going to switch gears briefly and talk about some examples from work looking at interactive effects of multiple stressors. Before I jump into this, I should define those interactive effects that I mentioned. So this is based on some work that was done led by Caitlin Crane in collaboration with Ben Halpern. And I'm showing you some hypothetical data that defines how we're using additive, synergistic, and antagonistic a little bit more carefully. So if you imagine this as a factorial experiment where you have a reference or control response, the response when the organism is exposed to stress A, stress B, and then you have 3 possible outcomes, which are shown in the colors. So an additive outcome is simply when the response to stress A and stress B are summed up. When the response is more pronounced than you might expect based on that additive example, you can call that synergistic, and vice versa for antagonistic. Now there's lots of issues here that I'm happy to chat all about, but I'm gonna move us forward. So the results from a meta-analysis that we did a few years ago now, and this was looking at any stressor in the ocean, and its effect when it was combined with another stressor, was broken down into those 3 categories. And so this is actually a different figure than what I showed you at the intro, which was just temperature and warming and acidification effects. But what you'll see again is that we see kind of the full range of interactive effects fairly evenly split. And as someone that's really interested in prediction and scaling up, this is not very useful. And it's even less useful when you look at it this way. So now I'm going to break this down into stressor pair combinations, again, using those same colors to identify additive, synergistic, or antagonistic effects. And what you can see here is that across really the broad range of stressor pair combinations, we see all 3 types of interactions. So it's not something that's really driven by a particular stressor pair combination. Now, this work has spawned a lot of really useful and interesting studies. I'm highlighting one here by Jeremy Peugeot and colleagues that came out a few years ago, where they suggested that we might consider a broader range of definitions for interactive effects. So different types of antagonisms and different types of synergisms based on the directionality of those individual stress effects. To a paper that just came out last week, and this was published by Caitlin Fong and colleagues, where they actually showed that depending on what you use as your control in your meta-analysis, you can change the breakdown or what you actually determine is the interactive effect. And so in this example, whether herbivores were absent or present was the control, dramatically really changes the outcome of that interaction. So this, I'm not going to solve this problem today, but I'm going to show you one way that we have been trying to approach this in my lab. And this is work that's being led currently by my graduate student, Emily Donham, where we're trying to embrace that complexity and lean into what we know about nonlinearity in nature. And we're doing this by focusing on one example, again, on this temperature and acidification, or temperature and CO2 interaction, in part because we have a really good understanding of the nonlinearity associated with organism responses to temperature. Okay, now one thing I'm going to point out that I think is kind of interesting here is when we finished polling the data in 2007 for this study, there were 3 studies that had measured the effects of warming and acidification together on marine organisms, and we're now, somewhere near 500. So there's been a really dramatic increase in this research. Okay, so as I mentioned, this is a thermal performance curve. We have a really great understanding of how physiology or biology responds to temperature due to decades of thermal physiology. So generally, we're going to see warming or performance increase with warming up to some thermal optima, beyond which you have a really steep decline in performance caused by enzyme degradation or other processes. We don't have a very great understanding of the response surface or response curve of acidification, but our best theory predicts that ocean acidification will shrink the thermal performance curve. So you'll expect to see also a nonlinear response still to temperature, but that will be limited somewhat by oxygen supply in an acidified ocean. Again, I'm not going to go into much detail here. The key thing that I want you to get here is that rather than just generalizing, if we rely on more nuanced theories, it might provide more insight into the patterns that we're seeing. Okay, so for this one example, I'm going to show, I'm synthesizing factorial experiments. And so you might, I have some hypothetical examples up here of what a factorial combination of treatments might look like. So your control in your warming only treatment would fall on that control thermal performance curve. And then your OA and your warming and OA combined, OA is ocean acidification, would fall on that acidification curve, thermal performance curve below. Now what we've done in this analysis is we're looking at the slope of the effect of the warming only treatment and the slope of the warming treatment on that acidified thermal performance curve. So this is how we're going to kind of I'm going to lay out the data. So the slope of the thermal performance curve is on the x-axis. The slope of the thermal performance curves in acidified condition is on the y-axis. Again, this is a little detailed, so you don't need to follow every little nuance of what I'm talking about here, but we're generally expecting things, the slope to be positive in our data. We might see some linearity, or we might expect some linearity where when the slope is positive on the thermal performance curve, you would expect it also to be positive on the ocean acidification thermal performance curve, but where that slope is negative, so over the thermal optima, you might expect it to be an even steeper decline on that ocean acidification thermal performance curve. We don't expect to see something like this. This is not really something we're looking for. Okay, so here's the data right now for all invertebrate species. And this is really the one figure I'm going to focus on for this really short talk. And a couple things you'll see. So the slope is highly significant. It is positive, but it's not one-to-one, suggesting that acidification might be changing the shape of that thermal performance curve to a certain degree. If we break this down into quadrants, you'll see that in these two upper and lower quadrants, generally the slope is in the same direction. It's either positive on both thermal performance curves or negative on both. And then it highlights some really interesting outliers. So some where the slope is positive in just the warming thermal performance curve, but negative on that acidified thermal performance curve, suggesting that acidification might be pushing the performance over that hump or the thermal optima. And then the opposite, where actually the ocean acidification is reversing that temperature effect. So it might be shifting the thermal performance curve to a certain degree. Okay, so I have lots of conversation I'd love to have with people about that. But what I really was trying to do was just provide an example of how I think we can use theory to actually drive our understanding a little bit farther. And in this context, we can use the information we know about the response curves, that they are nonlinear, to provide more context in this interactive effects, rather than trying to come up with new effect sizes. In that, those results, again, feed back into our observations and our experiments, and that is really an iterative process that's going to move us forward. Okay, so coming back to this big idea of scaling up in global change biology, I hope the examples I've shown highlight again how this ecosystem of interactions between observation, synthesis, and experiments are really important, in that in our search for generality, that the complexity, the variability, and the transparency have really important roles in helping us scale up. So with that, I should thank my funders, as well as recognize all of you and my collaborators who have made contributions. And then I think we're saving my questions until after the next talk, but Heather or Courtney can clarify.
That's right, Kristy. Thanks so much. We are saving questions to after the first two speakers. So really encourage everyone to use the chat or Q&A functions to log those questions. Because of the webinar format, we can't open up the conversation to every participant in this format. So you'll need to log your questions through that, and then we'll share them with the speakers. So thank you, Kristy. That was really interesting, and I think a great perspective on how we can use synthesis as a tool integrated with other modes of research to really better understand the world we live in. So thank you for that. I'd like to now introduce Dr. Patrick Gonzalez from the University of California, Berkeley, and also the U.S. National Park Service. He's going to discuss a global example of synthesizing evidence of anthropogenic climate change, examining wildfire, tree mortality, and biome shifts to guide biodiversity conservation and carbon emissions reductions. So Patrick, please take it away.
Thank you, Ben, and thanks to NCEAS and everybody participating today for your interest. Can you see my screen and hear me?
Yes.
Okay, great. So on the floor of Yosemite Valley, I woke up on a snowy Christmas morning 33 years ago. But since then, published scientific research has found that human-caused climate change, anthropogenic climate change, has altered ecosystems and human well-being around the world, including Western North America and Yosemite. Anthropogenic climate change has increased wildfire and tree mortality and shifted plant and animal species upslope. Now, today, I will examine climate change and the role of synthesis in answering these three questions on climate change impacts, conservation options, and carbon solutions. And first, the impacts, and I'll focus on three of the most substantial impacts to ecosystems: wildfire, tree mortality, and biome shifts. Now, cars, power plants, and other human sources pump twice as much carbon into the atmosphere than ecosystems can naturally absorb. And this is the fundamental imbalance that causes climate change. The emissions have intensified the greenhouse effect and increased temperatures globally to their hottest level in 8 centuries. And in the United States, colleagues and I have analyzed climate change at 800 spatial resolution across the country and in all of the 423 US national parks. And we found that climate change increased temperature in the national parks at twice the rate of the country as a whole. And this disproportionate exposure of national parks derives from their location in extreme environments. The Intergovernmental Panel on Climate Change has analyzed all of the potential causal factors, and this is in effect a synthesis of different data sources on different sources of heating of the earth, compiled and assessed in one common variable, and that's the flux of energy at the ground. And 0 here is pre-industrial, 1750, and bars to the right are heating. This analysis shows that human activities have caused almost all of the measured heating of the earth. Now, wildfire is a natural and necessary process in many forest ecosystems, in many forest grassland and woodland ecosystems, including the giant sequoias in Sequoia National Park, where fire managers burn intentionally using prescribed burning to reduce fuel loads and to simulate natural burning. But many agencies in temperate zone ecosystems around the world have suppressed all fires, even natural ones. This has caused an unnatural buildup of fuels in many ecosystems. Tropical forests have the opposite problem, regrettably. Intentional burning to clear land for agriculture and livestock causes most of the wildfires. So colleagues and I analyzed the patterns of burning and the change of fire frequency over the 20th century. And we see here that in temperate zone ecosystems, the temperate zone ecosystems have not shown a long-term change in fire frequency, but tropical ecosystems have. And this is not climate change. This is actually a result of human local change. So discriminating between the effects of anthropogenic climate change and other human factors requires these two scientific procedures of detection and attribution. And attribution derives from a number of different methods. We don't have time to go into all of the details. I'll leave this up for a moment for you to see. I'm going to show you now published field examples of increases in wildfire detected and attributed to human-caused climate change. Climate change has doubled the area burned by wildfire across the western US over natural levels. The combination of the yellow and the orange is the actual burning since 1985. And if you remove the climate change signal, the natural burning is this yellow curve. So climate change has doubled the area burned across the western US. Climate change increased burned area an astounding 7 to 11 times in British Columbia in 2017. This attribution compares the post-warming fire probability curve versus the pre-warming probability curve. And the climate dominated all other local human factors like ignitions from roads and all other physical factors in explaining wildfire in US and Canada national parks and protected areas since 1984. This is a synthesis of cases of attribution of increases in burned area around the world as part of my work for the Intergovernmental Panel on Climate Change Major Assessment Report that is in progress for publication late this year or early next year. And I've just shown you the 3 cases of detection and attribution denoted by the red circles. And what we see here is that in many other parts of the world, we only have detection, and in many cases, particularly in tropical ecosystems, deforestation, agricultural expansion, and other factors exert a greater influence on burned area than climate change. A self-reinforcing feedback of climate change, increased wildfire, then emissions of carbon, which feed back into causing climate change, occurs. And colleagues and I found that in California, wildfire, the 6% of the land that burned caused 2/3 of the emissions and caused California ecosystems to become a net emitter rather than a storer of carbon. Now, turning to tree mortality, across the southwestern US, anthropogenic climate change has caused the most severe drought since the 1500s. And a combination of the drought, the increased wildfire that we saw, and bark beetle infestations, through those, climate change has doubled tree mortality across the western US since the 1950s. In the interior west, the northwest, and California. In the West African Sahel, anthropogenic climate change has caused the death of 1 out of 5 trees and the local loss of 1 out of every 3 tree species. This is in an area that it has experienced the most severe decline in rainfall in the global instrumental record. And some more details on this, the tree density in 1954, about 10 trees of height greater than 3 meters, 10 trees per hectare on average, reduced to about 8 trees per hectare by 1989. So 1 out of every 5 trees died on average, and in some areas, the red areas, over 3/4 of the trees died. This is to give you an idea of the area. This is the area in Northwest Senegal. And this is the 9-county San Francisco Bay Area. And then you can see the area is greater than the area of Rhode Island and Eastern Massachusetts. So a pretty extensive area of tree mortality. And the attribution here was a multivariate statistical analysis of 215 different climate, physical, and socioeconomic factors. This is a synthesis of drought-induced tree mortality led by Craig Allen and updated 3 more times since then. And drought-induced tree mortality has been widespread around the world. 3 of these cases attributed to anthropogenic causes. And now turning to biome shifts. A biome is a major vegetation zone dominated by a distinctive plant form, and temperature and precipitation differentiate global vegetation into biomes. And actually, they're not distinct lines, they're clouds here, as you see in this depiction on the right. So a change in temperature or a change in precipitation can shift the biome of an ecosystem. On the eastern side of the Sierra Nevada in California, temperature differentiates vegetation into 5 different biomes going up the side of up to the tallest point in the lower 48. So this is a synthesis of biome shifts around the world, and it includes field cases of relatively long-term analyses, 30 years or greater. And this was originally, colleagues and I produced this, and it's been, and then I updated it for the Intergovernmental Panel on Climate Change. And we see that climate change has shifted biomes at sites in boreal, temperate, and tropical ecosystems. And again, the cases were at least 30 years, and some of them span more than a century. And we can see that increases in temperature or decreases in precipitation cause the biome shifts more than other human factors. This has occurred in Yosemite National Park, where climate change has shifted subalpine forest upslope into subalpine meadows. And in New Zealand, where climate change, in the Fiordland National Park in New Zealand, where climate change has shifted southern beach up into subalpine shrubland. And going back to West Africa again, this series of 2 slides shows the shift of the Sahel savanna and the Guinea tropical forest biomes. And this is 1945 and 1993. You can see the darker browns are a higher fraction of arid species, arid Sahel species. And the darker greens are a higher fraction of the moist tropical forest species. And you see before and after a shifting of biomes of approximately 30 kilometers. So what are the implications of this? Where should we prioritize biodiversity conservation under climate change? Now, this actually requires more analyses and basically modeling of projections using the IPCC emission scenarios. And this type of analysis indicates that climate change could increase wildfire frequencies up to 50% across a third of terrestrial land by the end of the century. Now, the conservation response to this is prescribed burning and letting natural fires burn. This can reduce the risks of high severity fires. But this is a response in those temperate zone areas that I talked about that have experienced this unnatural buildup of fuels. In tropical forests, the obvious solution is halting the use of fire to clear land. And many tropical forests, like this moist, humid, closed canopy evergreen forest in the Amazon, outside of a national park in the Amazon, are ecosystems that are neither adapted, they're not adapted or tolerant of wildfire. So colleagues and I, that was wildfire, to examine biome shifts. We conducted an analysis of different data sources to examine exposure, sensitivity, adaptive capacity, and the results were the vulnerability of biome shifts just to climate change, and then combining that vulnerability to climate change and habitat loss. This is the biomes, the global biomes, based on the baseline vegetation, the baseline climate, sorry, from 1961 to 1990. And then these are the biomes under the worst case, any change in 9 of the general circulation model IPCC emission scenario combinations. So this is the baseline, and this is today, and this is with climate change. And so you can see potential shifting of hundreds of kilometers, 1/8 of global terrestrial land at high risk of these shifts. And then when you include habitat fragmentation, the vulnerability increases to about half of terrestrial land area. So this all leads to a major question, and what magnitude of carbon emissions reductions would avoid these global ecological disruptions. And so this is a synthesis of syntheses, and using the burning embers method of the Intergovernmental Panel on Climate Change. And basically we have the four major ecological impacts here: biome shifts, tree mortality, wildfire, carbon loss. And this is the temperature increase relative to pre-industrial. And so you can see here, today is this small line here, and the Paris Agreement goal is a goal to limit warming between 1.5 and 2 degrees above pre-industrial. And of course, the darker the color, the more severe the concern. And all of that material, all of those syntheses of historical evidence go into determining this lowest transition from undetectable impact to moderate impact. So from white to yellow. All of that historical information goes there. And then the second part of the presentation, those future analyses, those future projections go into the other 2 transitions, from yellow to red and red to purple, moderate to high, and high to very high. So this type of synthesis was exactly what was used by the UN Framework Convention on Climate Change to develop the 1.5 to 2 degree goal of the Paris Agreement. Basically, it was to avoid these red areas. What level of temperature would avoid these red areas? So taking then that temperature threshold allows us to back calculate how much of our carbon budget is left, and basically, regrettably, at the current emissions, only 10 to 20 years are left to limit our temperature increase to 1.5 degrees. So that's the bad news. The good news is that we are making progress, and as you know, President Biden is returning the United States to the Paris Agreement. And even under the previous administration, which denied science and blocked action on climate change, the 24 states of the US Climate Alliance and Puerto Rico cut greenhouse gas emissions 16%, on track to meet the Paris Agreement goal. And in California, we cut our greenhouse gas emissions below 1990 levels, and so meeting the state target 4 years early through renewable energy, like this solar panel on a building in Yosemite National Park. So personal action is essential to meet this goal. And just through walking, biking, public transit, you can cut your personal transportation emissions substantially. And for those who live in the San Francisco Bay Area, taking BART, the subway, reduces your emissions 80% compared to driving a car. And of course, halting deforestation could avoid 15% of global emissions. But the major part, 85%, requires emissions reductions through fossil fuels. So in conclusion, I've attempted to through this brief presentation, attempted to answer these three major questions, and the three major questions I posed. And first, that the attribution, the synthesis of attribution of ecosystem impacts identify the areas of the most severe concern. And then analyses of climate change and land cover identify the priorities of additional conservation. The syntheses of evidence actually inform the calibration of the analyses for the future projections. And then third, that synthesis of major impacts across around the world indicates how the magnitude of carbon reductions that we need. And again, the good news is that IPCC, a lot of what we see about climate change is bad news, and a lot of what I've shown you. But the good news is that the IPCC assessment of options shows that we can still limit global temperatures to less than 2 degrees with concerted global action. And so the future of synthesis is protecting our world for nature and human well-being. Thank you.
Great, thank you so much, Patrick. We have some questions already coming in the Q&A area. If you haven't asked a question and would like to, that's the place to do it. Some are being live answered, which is great. If we have time to return to those to share them with the whole group, we will, but you can also check there to see what the speakers are responding to in those questions. I'm just going to go in order that they were posted here. And I guess this is for both Kristy and Patrick. Your thoughts on, the question is, how do we leverage the issue that journals are less likely to want to publish the observations and experiments and favor large-scale meta-analysis synthesis, but synthesis cannot happen without the experimental observational work being published and available? Any reactions or thoughts to that?
Well, my initial thought is that Ben actually showed us that over 1,000 synthesis publications have been published, but I think an approach that is fruitful and that I'm actually suggesting through my presentation is aiming syntheses at these important global questions, and basing them on the individual observational publications.
I think I agree with that entirely. I would also add that in framing our meta-analyses, I think it's also important to, in the way that we write them, to be really clear that we can also do hypothesis testing with our syntheses and our meta-analyses, and that there are, sometimes they open up more questions than they answer. And so I think if you're really clear and transparent about that in the way that we write about them in our publications, then it should be pretty apparent. In my experience, it's not too hard to get your experiments also published. It is hard to get your negative experiments published, but that's a whole nother issue we can talk about.
Yeah, and this next question is sort of related. I think the person asks they point out they like the concept of synthesis feeding back onto observations and experiments to inform subsequent synthesis, but then this raises interesting challenges when dealing with, for example, long-term research. How do you best build on past data into the future as ecology of place and technological approaches to capture the data change? So thoughts or reactions on that?
Yeah, I think that might be a longer conversation. That's a really big question. And to tell you the truth, I don't have an answer right off the top of my head.
Fair enough. Patrick?
Can you repeat the essence of that question?
Well, I think it's trying to get at the idea that the interaction between observation, experiment, and synthesis is also faces the challenge of changing data. Because if you're informing, like especially with long-term research, if you're informing your empirical study design with what you're learning from synthesis or the empirical work itself, the data streams are changing, and how do you deal with those changes when you're trying to synthesize?
Yeah. If you're fortunate, yeah, a lot of my work is reconstructing past tree species distributions, and if you're lucky, the data was collected in a way that you can find a common variable. But if not, then it might not be possible. But I showed a few examples of syntheses where that were able to extract one common variable that was relatively less affected by changes in methods.
Yeah. Okay, thanks. Next question states, I'm curious about the different attributions of responses to climate change and local human causes. Are they really separable cleanly? It's a framing that puts more direct responsibility for environmental change onto human action in tropical, often poorer countries than in temperate, often richer countries.
So in attribution, the most robust attribution assigns the fraction of the cause to the different factors. So it isn't just 100% anthropogenic climate change and 0% others. The examples that I were showing, anthropogenic climate change was the leading factor and dominated other local human factors. And I came back often to the problem of deforestation in tropical countries, really to highlight that this is an actual problem that, climate change exacerbates it, but the options for solutions are more clear cut and more immediate. Not everything is climate change, but of course the interaction of climate change and local human action makes it much more severe.
Thanks. Kristy, anything to add to that, or? Okay. All right, I think this is a question directed to Patrick, probably. Many of these studies considered singular mortality events. What is the strongest line of evidence, or for example, methods used, provided by these studies that directly links mortality to anthropogenic climate change? Or rather, what evidence is generally required to prove climate change impacts on mortality?
Okay, here again, I had a slide there of like 7 different attribution methods, and perhaps the strongest evidence for tree mortality comes from multivariate statistical analysis of all the different factors using a spatial analysis. And probably one of the more robust tests is canonical correlations analysis, where you can have multiple dependent variables as a function of, in that one case, I had 215 independent variables. Now, another method, and that would be the strongest if you have the data on the change in temperature and the change in precipitation, change in climate water deficit, but also the change in population, the change in livestock use, and then changes in soil and hydrology. And so that's a synthesis across a lot of different, mainly scientific disciplines, but actually socioeconomics, since a lot of tree mortality and wildfire is caused, is caused by people. So you need to, obviously you need data on human ignitions, and then for tree mortality, the effect of that densification of forests. So anyway, yeah, so the multivariate statistical analysis.
Great. Okay. I'm gonna jump to a question that just was posted that asks, what is the intersection between synthesis and prediction? And I'd also maybe throw in there, like, is there something different that synthesis can or can't do with respect to prediction versus other methods that we use? Maybe I'll start with Kristy and then we can go back to Patrick.
Yeah, so I was in the prediction chat room this morning, and it was perfect because it actually was really closely aligned to what I was interested in talking about, and it intersects with this question as well. I think synthesis has a really important role to play in prediction because the area of inference is so much larger. With being an experimentalist, I can do an experiment on sea urchins and acidification, and all I can talk about are sea urchins and acidification, right? But by doing synthesis and working across a number of experiments or a number of species, then I can actually talk about those rates and processes. So I can try to move away from that individual urchin and say, like, well, do we see generality across the wide range of taxa and events that we're talking about? So I think that's kind of addressing your question to a certain extent, Ben. I think that synthesis plays a really important role because we have a much bigger area of inference that we can work in. And I mean, I think of my syntheses often as building hypotheses, but I don't think that they're the answer. I think that for me, they build the bigger hypothesis that I can then go back to try and test.
Patrick?
Thanks. I'll just add to what Kristy said briefly that I tried to show cases where syntheses of historical changes could be used to calibrate the models for future projections under climate change. Yeah, so we'll leave that there.
Great. Okay, we have just a few minutes left before our break. I'm gonna put this, I think, last question. I think it's an important one to raise that was just posted, that both speakers were able to do synthesis using data collected over broad geographic scales, time scales, and collected by many groups. Many people don't want to share data because they feel they are often not invited to such synthesis efforts. How can we address this so people feel they are contributing to a larger goal, and how do we make the process more inclusive? Any thoughts on that?
Thank you. I have a couple thoughts. So there's been some thoughts actually published in the literature about this that are interesting, and probably Ben or someone at NCEAS could provide the links. You probably even know them better than I do. But there's this idea of creating different types of author lists. So for any given synthesis, could you have a data author list? And so those actually those people who contribute data to the synthesis get recognition that they can put on their CV, that this is, they have contributed to it. If the CV is the thing that is the, of value for that given person in their job or whatever. But I think moving towards that would be really helpful. I would love to be able to give everyone authorship on these meta-analyses, but then you're talking about hundreds of authors, and really it's a huge undertaking to do the synthetic work yourself. So there's a difference between the contributing data to actually doing the analyses. So I really like this idea of creating different author lists that could actually be recognized by the Academy or other workplaces.
Well, yeah, and in that line, I mean, that's the DOIs for data. Like if you archive your data and give it a DOI is one way to get credit for that contribution. It's cited that way, and you can report it in your citation metrics just like you do for publication, and you list it on your CV as a publication. It's a data publication. So certainly the field is trying to develop mechanisms for supporting that. I'll just add in my 2 cents. I think it's, there is a strong movement towards open data and open access to democratize science and make things more available to not just the wealthy institutions and countries, but to everyone, but also just to leverage the investment in gathering these data. And NSF is moving in that direction for requiring people who get funding from NSF to archive their data and so on. And I think my hope is that as we move into eventually a fully open data system, we have solved some of these other problems that come from attribution and credit and engagement of folks in these processes, because all data are available and we've built a system that supports that. Maybe that's Pollyannic and hoping for a bright future, but I think that's where we're headed. And I think, but for synthesis to feel inclusive, we need to address not just these data issues, but all sorts of other questions about, or issues about, how we make the process of doing synthesis feel welcoming to all people. And that's definitely part of what we're going to talk about in the next 2 days, and a larger task ahead of us in our field. So thank you for that question. And thank you to both our speakers, Patrick and Kristy. It was a great opening session for this. We are at break. Everyone, please come back by 11:15. But go stretch your legs, get a coffee or a drink if you're in Europe, or take a nap if you're a little tired, but set your alarm, come back, because we're going to do breakout sessions and really start to dig into some of these details. Heather, any parting words before break?
Yeah, just one logistical. So we will be back here in the main room after the break. And then we will be moving into the individual breakout rooms. So when you come back, just come back and join the main room once again. We'll see you at 11:15.